Okay, so today we're going to talk about molecular paleontology. So that's resurrecting sequences from 4.2 billion years ago. So this is history that we're relating, ancient history, recorded and preserved in genetic code. And it's a story of art, history, biology, and coding. Some of this is Chimera X art. That's a type 2 tRNA. That's a type 1 tRNA. So we're discussing origin of life and astrobiology, epigenetics. So tRNA modifications are very similar to epigenetics, and epigenetics is a major feature of human health. Discuss amyloids. So amyloids can be bad. They can cause things like Alzheimer's disease, Parkinson's disease, but they're also good at the origin of life. Without amyloids, we probably wouldn't be here. Transfer RNA, that's the meaning of life chemically. mRNA, when you think of mRNA, think of all the vaccines that keep you from getting flu every year. Transfer RNA is older than T-Rex. This is a living fossil and it's better preserved than T-Rex, which is just bones now. Bones turn to rock. Coding. Coding is the secret to biological complexity. So the genetic code, you have to evolve a genetic code to have biological complexity. Molecular paleontology. This is from 4.2 billion years ago. It's a story of art, history, biology, coding. So life evolved around tRNA, that's transfer RNA, and Zach Burton, retired professor from Michigan State University, and my collaborator is Lei, a professor at the University of New England, the Biddeford campus. So the origin of life was recorded in transfer RNA sequences, and here are the sequences. We'll discuss those in more detail later. And here's a three-dimensional representation. Genetic coding evolved first in transfer RNA and then was transferred, co-evolved into messenger RNA, and then stored in DNA. So in terms of the origin of life, think about coding first in tRNA, secondarily in messenger RNA, and then stored in DNA. Because of Watson and Crick, we think of it in the opposite way. We think of the genetic code in DNA and then being made into RNA and then being made into protein, and tRNA is part of protein synthesis. But in terms of evolution, think about it in the um, other direction. So this is a ribosome um, from a bacterium, and it's sort of a molecular horror, a very complicated assembly. But if you strip it down to its component parts, you see messenger RNA, you see an A site tRNA, a P site tRNA, and an E site tRNA. So this is the exit site. This is the peptidyl site where the protein chain spends most of its time. And this is the aminoacyl site where an incoming amino acid um, gets added to the chain. And when it gets added to the chain, the peptide chain here gets transferred over to there. The A site tRNA becomes the P site tRNA, and the E site tRNA gets booted. This becomes the E site tRNA. So it's an assembly line for synthesizing proteins. The ribosome is a molecular horror at this level, but at its core, it's a simple assembly line for building proteins. So the code, the genetic code, is limited in tRNA by wobbling. And so just a few notes about what wobbling might be. So this is a wobble base right here. Watson-Crick, Watson-Crick, wobble, Watson-Crick, Watson-Crick. So these are two different representations of this loop here. And at the wobble position, there's only pyrimidine versus purine recognition, which means there are only two possibilities at this position, four here and four there, but two there. So the code is limited by transfer RNA, by wobbling. So wobbling is a feature, perhaps not surprisingly, of the tRNA wobble position. So that's the wobble position. Watson-Crick, Watson-Crick, wobble, Watson-Crick, Watson-Crick. At a Watson-Crick position, you have four possibilities. That's A, G, C, and U. A, G, C, and U. And there are two possibilities here. At the base of the code, this is, if it's a purine, it's always G, never A. And if it's a C or a U, it makes no difference. It encodes the same amino acid. So the complexity of the code then is two possibilities here, four there and four there. That comes out to 32. 16 times 2 is 32. In school, you'll learn that the complexity of the code in DNA and messenger RNA is 4 times 4 times 4, and that's true. 64 possibilities, but only 32 possibilities in tRNA. Wobbling is a feature of the tRNA wobble position. The code is limited in transfer RNA. So at a wobble position, only purine versus pyrimidine complexity is achieved in evolution. 2 times 4 times 4 is 32. 32 assignments. Purine, pyrimidine, wobble, Watson-Crick, Watson-Crick, AGCU, AGCU. So complexity 32. But it's worse than that. The genetic code froze at 20 amino acids plus stops. And it did so because of fidelity mechanisms. So translational fidelity mechanisms cause the code not to give you your full 30 amino acids that you would have deserved based on the complexity of tRNA. The genetic code initially froze, however, at 8 amino acids. And the reason for that is because originally there were two wobble positions. So what am I saying here? I'm saying that this was always a wobble position and will always be a wobble position. And this used to be a wobble position, but wobbling was suppressed at this position. And you can only read one wobble position at a time. So the complexity of the code with two wobble positions is 2 times 4 or 2 times 4, depending on whether you pick this as your wobble position or that is your wobble position. So that's 8 amino acids. So the code freezes initially at 8 amino acids because of wobbling in tRNA. So this is the solution to the puzzle, and it may seem a little daunting. It took me a few months to work this out, and there won't be a quiz at the end, but basically, these are the linear sequences. This is for a type 2 tRNA. This is for a type 1 tRNA. This is a two-dimensional representation of a type 1 tRNA. And this is a two-dimensional representation of a type 2 tRNA. So these are real tRNAs from real living organisms. The difference between a type 1 tRNA and a type 2 tRNA at the base of the code is only the variable arm here, which is 7 yellow and 7 green, versus 5 yellow here in the V loop. So these 5 yellow correspond to these 5 yellow. So if you delete 2 yellow and 7 green and link this up there, you get this. So an early version of this molecule was processed into this molecule here. Here it is in linear form. So the precursor to type 2 and type 1 tRNA was 331 nucleotide mini helices ligated together. So if you cleave this, this is a precursor to type 2 and type 1 tRNA. If you cleave it here, there, and there, and there, you get 331 nucleotide mini helices. But if you ligate them together to replicate them, they can then be processed into type 2 or type 1 tRNA. And to process to type 2 tRNA, you make a 9 nucleotide deletion. This is 7 nucleotides. That's 7 nucleotides. 7 plus 7 is 14. If you delete 9, you're left with 5. And those 5 are these 5 right here. And the same deletion in type 1 tRNA. In type 2 tRNA, there's no deletion in the V loop region. So 7 yellow plus 7 green. And these 7 yellow relate to those 7 yellow, and those 7 green relate to those 7 green. So that's here, 7 yellow, 7 green. And if you want to process this molecule into this molecule, you delete 2 yellow and 7 green. That's this deletion right here, leaving you with CCG, CC, CCG, CC. So if you look at the sequences, this is a GCG repeat, this is a CGC repeat, this is a UAGCC repeat, this is the same as these 5 right there, a little bit of a sequence rearrangement to make a better D-stem. And here, CCG, CC, CCG, CC, right there. So GCG repeat, GCG repeat, CGC repeat, CGC repeat, CGC repeat, GCG repeat, GCG repeat, GCG, CGC, UAGCC repeat. This stem loop stem, that stem loop stem, and this stem loop stem, this stem loop stem are originally identical sequences. The sequence changes when you fold into tRNA. The T loop, this is called the T loop sequence. This is called the D loop sequence. The D loop interacts with the T loop. And so there's a little bit of a sequence change here to support the tRNA fold. You have, should be UAGCC, it's changed to UGGCC. It's a G because it intercalates there and it hydrogen bonds there. So A won't do that, G will do that, so that's why this change occurred. These sequence changes help to form the elbow. The elbow is where the D loop binds the T loop. Okay. So now we're looking at rather important intellectual property in coding, and we've looked at this before. This is a representation of this loop right here. This is a different view. And the reason this view is shown is to show you that the U-turn The U-turn is a U-shaped turn in the RNA backbone. So because there's a U-turn, this strand and this strand interact, and the loop is more stable than it would otherwise be. So there's a chemical selection to this loop and that loop in pre-life. This is the wobble position, Watson-Crick, Watson-Crick. We've discussed coding. Part of the suppression of wobbling at this position is the modification of this position. So the ones in ball and stick are modified bases, and the other ones are unmodified bases. So modification of this base is part of the reading of that base. And so wobbling was suppressed at this position. This position was probably never a wobble position. And so four possibilities. This is a wobble position. So it's G if it's a purine, it's never A. And if it's C or if it's U, it's read the same. There are very few modifications to these bases. You can't have any modifications that affect coding because this is coding. That's the triplet code. Wobble, Watson-Crick, Watson-Crick, like that. If you wanted to suppress wobbling here, you can't modify that base because that's a Watson-Crick base. It's involved in coding. And if you modify this base, it's on the other side of the U-turn, so this base doesn't um, doesn't really respond to it. So there is no way really of suppressing wobbling, at least in an evolved system. So you may want to have 64 amino acids encoded in your genome, but you can't have that. Okay, so we're back to our ribosome again, and it's an assembly line for building polypeptide chains. And you can strip this down to its central core parts, which are tRNAs and messenger RNAs. And then you have a lobe here to make polypeptide chains, and you have a lobe here to stabilize and guide the messenger RNA. So you can simplify this and you can build simpler ribosomes. And you could go into your laboratory and take these plans and uh, use them to make polyglycine. Polyglycine is the money of the pre-life world. And I'll try to show you that that might be so in a slide or two. This is the most stripped down form of polypeptide synthesis. This is a more complicated form. These are all RNA components. And so you don't need much beyond RNA in order to make this work. This one's based on 31 nucleotide mini helices. It could also be based on tRNA. So what does polyglycine do in the pre-life world? The pre-life world is actually complicated, and I don't want to go through the complexity in much detail, but these are polyglycine amyloids. They're also membraneless organelles, which involve polyglycine and probably RNA components. And you can take polyglycine polymers and you can start to build a protocell around this core. So in a cell, the cell is packed with materials. And these are some of the examples. And polyglycine amyloids act like primitive enzymes. Membraneless organelles support pre-life chemistry. This is really a complicated assembly, and this is the background for evolution of tRNA, ribosomes, and translation systems in living organisms. So to evolve life, first evolve tRNA, co-evolve messenger RNA, stored the code in DNA. DNA is a stable storage form for the genetic code, but in terms of inventing the code, originating the code, it originates in tRNA, gets cast into messenger RNA, and then gets stored into DNA. And you have to co-evolve a ribosome, and we discussed that in at least a little bit of detail. tRNA evolution was solved as a simple puzzle. This could be done because tRNA evolved from RNA repeats and inverted repeats. So GCG, CGC, GCG, CGC, GCG, CGC, stem loop stem, that's an inverted repeat, stem loop stem, and this is a UAGCC repeat that also forms a stem loop stem, an inverted repeat. And so finding the pattern um, went a long way to figuring out the solution of the sequence. So when I started this, I thought this was an unsolvable problem. And very quickly, I realized that this was a repeat in the sequence. The stem loop stem was homologous to the stem loop stem. And from that, I figured out the rest um, of the puzzle. I didn't know I was working a puzzle when I started this. So we're back to this slide. Evolution of tRNA was from RNA repeats and inverted repeats, and ligation of 331 nucleotide mini helices. And you can think of this as the first theorem in biology. Biology doesn't have any theorems, but I would say that this is very close to one. A theorem is a proven model as opposed to a hypothesis. This is certainly stronger than a model or a hypothesis. Okay. There's no chicken and egg problem in evolution of the genetic code. And I don't know whether you agonize over this, but many people do. So the idea is that the genetic code can't evolve because it can't know ahead of time that it's going to be this complex. But the answer to that is that it starts out making dumb polymers, polyglycine, the money of the pre-life world, and then glycine, alanine, aspartic acid, valine, the four simplest amino acids. And then you add leucine, serine, glutamate, and arginine. Now we hit um, uh, we freeze the code at 8 amino acids, and we have to break that frozen code. Uh, if you have aspartic acid, you have asparagine because you can have tRNA linked chemistry to change aspartic acid into asparagine using an imidotransferase. And if you have serine, then you can convert it into cysteine using a cysteine synthase. And then if you have glutamate, you can change it to glutamine using an imidotransferase, which can start out being a ribozyme and then become a protein when you encode proteins. When you have this level of complexity, you can make your first proteins. And then you go to 20 amino acids plus stops. And so there's a lot of innovation along this line. But as you get a more and more complex code, you've developed a whole bunch of intellectual property that needs to be protected, and it gets protected by fidelity mechanisms. And that's maybe a talk for another day. But um, you have a war between innovation and air catastrophe, and fidelity mechanisms co-evolve along with genetic complexity. So what have I told you? I've told you a story about the origin of life. In fact, I've told you the main thoroughfare of the story. Evolution of tRNA is the story of the origin of life and complex life. And it's a story also of astrobiology, because if you want to go to Saturn's moon Enceladus and invade and evolve life there, you'd better do it pretty much the way we did it here on Earth. If you try another means, I'm not sure it's going to work. I, I'm not certain about that, but that's something to consider. May have to work in an aqueous environment with RNA as the basic uh, coding material. Story about epigenetics, an important issue in human health. tRNA modifications are 4.2 billion year old epigenetics. You can think about them that way. Amyloids are important. Amyloids were critical at the origin of life, as I tried to indicate in one of my slides. Uh, as you get older, you worry about Alzheimer's disease, Parkinson's disease, other amyloid diseases. Um, but membraneless organelles are important for human health and human cell homeostasis, and those can be misregulated in order to give rise to amyloids. So amyloids are important to the talk. tRNA is really the chemical meaning of life. Um, when you think of mRNA, think about the vaccines. It's important that you take your medicine and get as many vaccines as are offered, because that's the key to your health and the health of your society. Transfer RNA is older than T-Rex. It's much better preserved. It's a living fossil uh, of events 4.2 billion years ago. And coding and the genetic code, that's where biological complexity comes from. You don't get organisms like us without a genetic code. And molecular paleontology is the means applied here to a problem 4.2 billion years old. So the puzzle of tRNA evolution was laid down about 4.2 billion years ago, and it's been solved. It's been solved for all time on this planet. It's a story of art. I hope you like my art. History, biology, and genetic coding. So life evolved around tRNA, transfer RNA, and I'm Zach Burton, a retired professor from Michigan State University, and Lele is my collaborator, professor at University of New England. Thanks so much for your attention.